I'd like to begin by wishing you a belated happy 30th anniversary to the Galiano Conservancy. Maybe 3 years belated, I suppose. But what a remarkable list of accomplishments for your conservancy. And it's been my great good fortune as one of the early members of the Habitat Acquisition Trust to work with early efforts of the Galiano Conservancy, probably almost 30 years ago, I suppose, with this rascal, who is a good friend. And we worked together when I was chairing the board for the Habitat Acquisition Trust on projects like the Great Beaver Swamp in Lachlan Lake, and especially on the community forest and the early management plans for the community forest. So I have very fond memories of working with Ken on these projects. I was asked today to talk about mushrooms of British Columbia. As Risa mentioned, there's a new Royal BC Museum handbook that came out in September of last year on the mushrooms of British Columbia. Although I've done a lot of work with, well, with some people on this call, even with plants, and I've written a lot about plants. I actually did my master's in fungi back in 1982 at the University of British Columbia with Robert Bandoni. So I'm really coming back to my training, getting back to working more with fungi, both in research, in teaching, and in writing here. The Royal BC Museum Handbook series, of course, has a lot of wonderful books, many of which I grew up with, about different taxonomic groups in British Columbia. They even have previous versions of mushrooms of British Columbia. The Mushrooms and Other Fungi of British Columbia by George Hardy in 1946, and Guide to Common Mushrooms of British Columbia by Robert Bandoni, my master's supervisor, and Adam Shavinsky, first published in 1964. That's a long time ago. Give you an example of how long ago that was. That's the beauty of being quite old, is you can you can have been at things for quite a while. The 1976 second edition of Mushrooms of British Columbia was the first museum handbook to feature the stunning new development, color photographs. So that was quite a while ago. I apologize if you hear a bit of clicking when I'm speaking. I'm sucking on a cough drop. I just tested positive for COVID this morning. So you can be very happy that you're experiencing me in a virtual meeting. And if you hear a little bit of clicking, it's just cough drops. My apologies. Mushrooms are fungi. Fungi are one of the 3 large kingdoms on Earth. There are additional kingdoms beside the large ones: plants, animals, and fungi. When I was at university, some of the textbooks still treated fungi as sort of degraded plants. And fungi are in a lot of universities are still taught in the botany department, but they are not plants, they're not animals, they are a different kingdom again. Pay no heed to this, except to notice that if we look at a chart showing the evolutionary and genetic history of different groups of organisms, the plants are in the top left, the uniconts or opisthocons, which includes animals and fungi, are in the bottom right, and the other kingdoms are arranged like this. We won't dwell too much on it, but just to make the point that the fungi are very closely related to animals and not at all related to plants. This is not the way some people see the world, I know, but one way of thinking about it is that when you see a mushroom in your yard, that mushroom is much more closely related to you than it is to any plant. Right? That's the nature of things. Fungi are eukaryotic. That means they have nuclei like plants and animals, unlike bacteria and archaea. They're heterotrophic, means they can't make their own food like animals, unlike plants, most of which are autotrophic. They're absorbent. They suck up their food from the environment. They can be various shapes. They're amoeboid or they're unicellular, in which case we generally call them yeasts, but they're usually made up of microscopic filaments, as in the bottom left photo here, about 5 microns in diameter, not something you can see with the naked eye. They generally don't move around very much. The cell walls contain chitin, which is something a number of animals, including most insects, do, and plants don't. And most reproduce by various types of spores in the bottom right there, which you could think of, if you like, as simple versions of seeds. All mushrooms are fungi, but not all fungi are mushrooms. So we tend to call macroscopic fungi, those that we can see with our naked eye, mushrooms. But the vast, vast majority of fungi are microscopic. And they're things like the yeasts at the bottom here that we use for brewing and baking, or microscopic filaments, the things that turn your cheese or your orange blue in color, tend to be species of penicillium, like the one shown in a micrograph on the top right, or the Penicillium roquefortii, which is growing through this cheese on the left. So these microscopic fungi are what make blue cheese blue. So anyone who's a fan of wine, beer, whiskeys, baked goods, or blue cheeses appreciates products of the fungi. So if we zoom in on these fungi, we see 5 major groups. And I'm going to go back a second there. The bottom 3 groups, probably most of you have never seen and will never see, and I'm not going to talk any more about. So just very briefly, the zygomycetes, and they're all named, like all plants, like all animals, like all fungi, by the way they reproduce sexually. Okay, that's how biologists organize living things in any group. Not because they're obsessed with sex, though some biologists are, but because the way you reproduce sexually tends to be evolutionarily conservative. That means it doesn't change much over evolutionary time, and so indicates long-term relatedness. Zygomycetes make zygospores, like the one shown here. They're generally decomposers, occasionally parasites. And the one on the left here, some of you may have seen it. It's microscopic, but if you see, if you have soft fruit like strawberries around for too long and they get too soft, they might grow a clear fuzz on top that's got black heads on it. That's thousands or tens of thousands of zygomycetes growing on it. Also fairly common for those who are fans of dog shit. You commonly see a white fuzz on dog shit, and that's decomposition by zygomycetes. Other than that, you're unlikely to see them. Microsporidia, like the one on the right there. Parasites of animals, you've probably got microsporidia inside you, but you're unlikely to see one in your lifetime. And the other group is the chytrids, Chytridiomycota. Generally decomposers. They're sometimes called water molds. You can isolate them from water by using seeds as baits. If you're really keen on going fishing for chytrids, that's a picture on the left of the world's most famous chytrid. You've probably never even heard of the world's most famous chytrid. That's Batrachochytrium dendrobatitis. And that's the species that's been implicated, perhaps, in global amphibian decline. So when you hear about chytrids named as potential culprit in global amphibian declines. They're talking about Batrachochytrium dendrobatitis and relatives. Other than that, you'll probably never hear of any of the species on this page and never see them again. So the rest of this talk is going to be about the ascomycetes and basidiomycetes. Mycetes just means fungi. So the ascomycetes are the fungi that make asci, and the basidiomycetes are the fungi that make basidia. Okay? Basidiomycetes includes just about anything you might think of as a mushroom. Here's the short-footed russula. If we look very closely at its gills, we'll see, and again, you need a compound microscope to see this. Well, in this case, you need a scanning electron microscope to see it. You'll see these sort of club-shaped structures with 4 pointy tips that have spores at the top. These are basidia. They line both sides of the gills, of each of the gills, and they produce the sexual spores. That's basidiomycetes. Almost everything you think of as a mushroom is a basidiomycete. A small number of them, oh yes, so there's a basidium, basidiospore. A small number of them are ascomycetes. The cup fungi, for example, are ascomycetes. So again, if we look at the surface using a compound microscope, we'll see these long sac-like structures. They're generally described as sac-like in books, which is not terribly helpful because they don't want to say it's shaped like a condom. But if you are prepared to accept that these asci are shaped like condoms, each containing 8 spores, that might be a good way to remember them. In this case, there are sterile cells, the ones staining golden in this picture in between them. So the sexual spores of the ascomycetes are produced in asci. In the case of all mushrooms, the mushrooms are the fruiting bodies. Think of them as being like apples on an apple tree or huckleberries on a huckleberry bush. So most of the fungus is in the ground or in the tree or in you sometimes. It's growing as so the basidia you can see on the right-hand side here under this mushroom are producing the spores. There's the spores in the bottom left. The spores grow into the filaments, which are called hyphae. If there's a whole bunch of them, they're called mycelium. And that grows through the soil and produces fruiting bodies when it wants to have sex. All right. And that's just as a simple example of that. If you have a favorite chanterelle spot on Galiano, you know that you can go back there the next year and there will be chanterelles there again because the fungus is always there in the ground. But we only know fungi are there for the most part when they produce their fruiting bodies. Okay, so that's what mushrooms are. It also means 2 other take-home lessons for me from that. One, that if you pick mushrooms without damaging the soil, it should be as sustainable as picking apples from an apple tree without breaking the branches. And the second is that the surest sign of true love is if someone shows you their mushroom patch. People bring each other flowers or cook meals or exchange golden rings even sometimes. But if things break down, all you've ever lost is a golden ring. But if you show someone your mushroom patch and subsequently break up, they will forever remember where your mushroom patch is. So if you're looking for a sign of true love, I think sharing a mushroom patch is the surest sign. There's a lot of details. We could spend the next half hour on this diagram, but I think that's enough for now. So what I'm going to do is very quickly move through some of the different groups of mushrooms here based on morphological features and give you some examples of things that you might find on Galiano Island. This is the front page from a free online exceptional program called MycoMatch. It used to be called Matchmaker, which I wasn't allowed to load on my government computer for many years because they suspected it was some kind of dating site. Now it's called MycoMatch and is a great way to identify mushrooms on synoptic keys. And it begins, as our book does, and a lot of other electronic and print copy resources do, by dividing the mushrooms up into these relatively easily recognized groups based on how they look. It's worth noting that some of these represent what a biologist would call a natural group. That means they're related to each other, and others don't. So a quick trip through these. Veined. A lot of these differences are based on how the mushrooms elaborate the surface that they have the basidia on. So you remember the basidia make the sexual spores. They're what mushrooms are all about. If the underside of this winter chanterelle or this golden chanterelle is simply a flat surface, maybe you could put, let's say, 10,000 basidia on it. But if you make ridges underneath, called veins here, or you make gills or pores or teeth, maybe instead of being able to put 10,000 basidia on the undersurface, you'll be able to put 100,000 or 1,000,000 basidia on the undersurface. And if your purpose of a mushroom is to produce sexual spores, if your purpose is to do that, then more basidia is better. And that's a biologist's interpretation of why mushrooms make these veins or ridges, as they do in golden chanterelles and winter chanterelles, or yellowfoot, if you like, or gills, or teeth, or pores, or other ways of making more surface area on the underside of a cap. So there's a number of the veined mushrooms where the undersurface is not gills. They're fairly blunt edged, fairly shallow, and they tend to branch or come together, as opposed to gills, which tend to be sharp edged, deep, and all parallel. They provide some of our favorite edibles. We'll talk a bit about how they grow later on. Well, so this presentation has two parts. I'm going to walk you through these different groups of fungi with a few examples in each case of mushrooms that you will find on Galiano Island. And this is to give you an idea of the sorts of diversity of form and the beauty of the fungi, of the mushrooms in British Columbia. And then I'm going to look at what those fungi are doing in ecosystems. That should take about 45 minutes and allow enough time, lots of time at the end for questions or discussion. About 2/3 of the mushrooms we call fungi, of what we call mushrooms, have gills underneath, and these are blade-like plates, and the basidia are arranged on either side of the gill. Again, there was a friend who was at a mushroom show and very bored, and he was pulling the gills off a mushroom and arranging them over the top of a folding table. And a relatively medium-sized mushroom like the clustered wood lover, if you take all of the gills off and put them on the top of a small folding table, say the kind you'd play cards on, they cover about half of it from a not very big mushroom. And remember, there's basidia on both sides of the gill. So the other half of the table would be covered too. So even a mushroom like the clustered wood lover probably has enough surface area on its gills to cover something, say, 30 or 40 times its size. And that's a huge benefit if you're putting your basidia on it. Lots of beautiful gilled mushrooms. Boletes are an awful lot like gilled mushrooms, but on their undersurface, they have pores instead of gills or teeth or veins. Here we have the king bolete, one of the most sought after mushrooms internationally and in BC, the porcini or the sep, the steinpilz. If a mushroom's got a lot of names internationally, you know it's delicious. But there are lots and lots of boletes out there. The pores, I like to think of them as sort of like toilet paper tubes, if you like. And so imagine the undersurface there. You've glued together, oh, I don't know, say 50,000 toilet paper tubes. And you can see in the cross section on the king bolete there where we've knocked it off, you can see how long these tubes are. And the inside of each of those tubes is lined on all of its surfaces with basidia. So imagine the huge increase in surface area from having pores. Some mushrooms have teeth underneath. Probably the most well-known example is the hedgehog in the top right there. And that's because people collect it for the table. It's much, I much prefer it to chanterelles, say. But it's the same basic concept. There are teeth, rather like icicles hanging underneath the caps, and the basidia are arrayed along the icicles. So everything you've seen so far is basidiomycetes, right? Some mushrooms just look like clubs and just have their basidia or acai over the surface. And here we have the purple fairy club on the left, and on the right, truffle eater. So that's a 2-for-1 photo. The spherical structures at the bottom are the deer truffle, which grows under the ground, which fruits under the ground. That's why it's a truffle. And you can see all those spherical objects are all covered in earth. They all would have been buried in the soil. And here they're being attacked by a parasite, the truffle eater, Tulipocladium, which is related to the zombie ant fungi. More on that later, but this is more of a zombie truffle fungus. The club-shaped fungi are just fungi that are kind of club-shaped. They don't represent any natural group. They represent basidiomycetes and ascomycetes, so fundamentally different. But from a field point of view, they're all shaped rather like clubs. I threw this one in just for fun. This is the dead man's fingers, and I think it bears a pretty close resemblance to its common name. Dead man's fingers, another club-shaped fungus. This one's an ascomycete. There are fungi, there are clubs that are sort of repeatedly branched and tend to look like marine corals, and they go by the name coral fungi. They're all basidiomycetes again, and they are mostly related to each other, but not all related to each other. Polypores, these again are all basidiomycetes, and like boletes, they have pores on their lower surface, but they are woody or leathery, whereas the boletes are kind of fleshy. There are some other differences, but one of the main one is that most of the polypores are wood decay organisms. And here's a couple of examples of that. One of them, the red-belled conch, BC's most famous conch, which probably no one's ever heard of. It's certainly the most common and abundant, widespread, and it's in the last few years been renamed. And I'll mention it here. I'm not going to dwell on scientific names, but this is Fomitopsis mountsi. Anybody who has studied or pursued mushrooms here may know it as Fomitopsis pinicola. Turns out it's a little bit different from the European species, so it needs a new name. And in this case, the new name is for not just a mycologist, which is unusual, or a BC mycologist, which is more unusual still, but for a female BC mycologist. So this is named for Irene Mounts. who in the 1940s studied the fungal decay of airplane-grade Sitka spruce in Haida Gwaii, at a time when the Sitka spruce was necessary for the war effort because it's what they made mosquito bombers out of. And after the war, went on to work at the research station in Saanich, I believe. And in the early 1950s, got married and then promptly lost her job because at the time, the federal government didn't employ married women. Bit of a career misstep for Irene Mounts, but she does have the red belt conch named after her. Here is the dyer's polypore. Also, if there's anybody here this afternoon who's into dyeing fabric with mushrooms and lichens, you may be familiar with the dyer's polypore. Very common root parasite. Very beautiful. A lot of fungi are gelatinous, like the witch's butter and the apricot jelly. And again, there's ascomycetes and basidiomycetes here with their fruiting bodies arrayed all over the surface of these lovely gelatinous organisms. The witch's butter is a good reminder that the fungi will oftentimes appear where you least expect them overnight. And so they have all kinds of names of witches and pixies and fairies and so forth associated with them. They're believed to be of supernatural origin. Puffballs. I think everybody is familiar with puffballs. Generally, they're almost without exception good edible mushrooms when they're young. And here's a picture of the Western giant puffball, which is known largely from interior British Columbia, but has one outlier. I've seen it as far west as the Pemberton Valley, but it has one outlier population in Metchosin, British Columbia, where I live, because this is really the best mushroom for breakfast that I know of. You can slice it, you can slice steaks off it so they fill your fry pan. And my oh my, I don't think there's a better breakfast than a nice fried giant puffball. Oh, sorry, all basidiomycetes. In this case, they produce the spores inside and have a tear or a natural opening where they puff their spores out. Bird's nest fungi, again, all basidiomycetes. Remarkable resemblance to little bird's nest. The eggs in the nests are clusters of spores, each containing millions of spores. And when a raindrop falls down into that cup, it blows these little clusters of spores, these little eggs, sometimes up to a meter away from these little cups. And the cups are, well, in this photo here, they'd be 2 to 5 centimeters diameter for the two different species. So blowing the spores a meter away is quite an accomplishment. Morels, and similar, are, I had a good morel feed the other night. Morels are among the favorite edible mushrooms in British Columbia, much more abundant in the interior than on the coast, alas, but they will pop up on the coast from time to time. And I suspect people on the call tonight will have harvested morels on Galiano Island. And then there's a lot of spring morels and false morels and the like, all ascomycetes with the asci arranged all over these surfaces. And the surfaces, again, are believed to be so elaborated so that it creates more surface area. Cup fungi are all ascomycetes, again, wood decomposers, and they have an upper surface lined with asci. And truffles is simply a generic name we give for fungi whose fruiting bodies are produced underground. Of course, there are famous truffles of commerce, like the Perigord truffle or the Italian white truffle. We have the Oregon white truffle, spring and autumn, two different species. Here in southwestern British Columbia, It's probably on Galiano Island. If it is, I would like to know. And it's a truffle of commerce in Oregon and Washington, where it's the basis of a thriving new industry. BC probably has 60 species of truffles, including the deer truffle and red gravel shown here. The vast majority of which are, they don't have the tastes and the smells of the commercial truffle species, and so aren't important as human food, but for some small mammals in the season when they're fruiting, they can make up more than 90% of the diet for flying squirrels, chipmunks, and so forth. So very important in natural systems, even though we're not eating a lot of them. And then there's some fungi that just form crusts. One of the most important is the lobster crust in the top right there. It grows over, in our area, it grows over the short-footed russula. So it's an ascomycete fungus growing over a basidiomycete mushroom and essentially obscuring the gills and turning it into something that doesn't look a lot like the mushroom that it started with, but the parasitized short-footed russula on the top right there is what we call a lobster mushroom, and is one of my favorite edibles. And I'm sure there are people here tonight who have feasted on lobster mushrooms. That's what a lobster mushroom is. It's a short-footed russula in our part of the world that's been parasitized by an ascomycete crust fungus. Okay, BC is large and diverse, probably more climatically and ecologically diverse than just about any area in the world at temperate latitudes. These are the biogeoclimatic zones. And that's why BC has more species of just about anything you can think of than any other province or territory in Canada. More bats, more birds, more plants, and more mushrooms. So we have records of about 3,400 species of mushrooms from British Columbia. Unlike some of these other groups, the mushroom composition, the mushroom communities in British Columbia are very poorly known. So we have records of about 3,400 species. I'll be surprised if there are less than 10,000 mushroom species in British Columbia, let's say. And there's certainly lots of new species on Galiano Island for anybody who's interested in looking for them. To protect mushroom habitat, which is what the Galiano Conservancy is all about. Did you know that's what you were protecting habitat for and restoring habitat for? It's for mushrooms. The best study that's been done in Canada on the importance of habitat was done in Clayoquot Sound. Over 5 years, they sampled 6 habitats: dunes, spruce fringe forest, old growth forest, second growth forest, bog, and estuary. They collected 551 species of macrofungi, keeping in mind that that's probably more than twice as many species of plants as you could collect in that area. 17 to 36% of the species in a habitat were found only in that habitat. Of the 551 taxa, only 28 were found every year, and 308 were found only in 1 year. Remember, they're just looking at fruiting bodies here. So that suggests that for a lot of the species, they don't produce fruiting bodies every year, or maybe not every decade. And if you want to do a good fungal survey of an area, you need to commit to, say, 10 years of sampling. And the old growth forest had approximately 4 times as many species as bog and estuarine habitats, and about 1.5 times as many species as the rest, which is to say, if you want to maintain fungal habitat, one of the most important things you can do as a conservancy, in addition to protecting whatever, well, not older forest, but mainly second growth forest with veteran trees in it, is available, and then leaving it for a few hundred years. And I know that's the plan. It's critically important for a lot of fungal species. There are some we only find in old growth forests. So, as if we needed any more reasons to protect our remaining old growth forests, do it for the fungal habitat. I don't want to go any further without mentioning people's fear of mushroom poisoning. This largely comes from the British Isles, is not something that's shared with a lot of other parts of the globe, and is grossly disproportionate to the actual danger involved. We tend to have a poor evaluation of risk for a whole bunch of activities. Searching for and eating wild mushrooms is one of them. There are poisonous mushrooms out there. You should be careful. You should know what you're harvesting if you're foraging for plants or for fungi. However, there aren't a lot of poisonous mushrooms and there aren't a lot of poisonous plants out there. Not really poisonous ones, I would say, the ones that would give you any more than a rumbly in your tumbling. As far as we know, in the recorded history of British Columbia, going back about 150 years, let's say, everybody think of a number. How many people have died from mushroom poisoning in British Columbia? Everybody think of a number. I'm going to let a growling dog out of this room and I'll be back in a second and we'll see how close you are. There you go. And that's right. The answer is 3 in 150 years. There are usually more people killed in a year by lightning than have been killed in 150 years of eating mushrooms in British Columbia. And there are certainly more people, way more people, that have been killed by eating poisonous plants in British Columbia than have ever been killed by eating poisonous mushrooms. So if you're foraging for anything, you should know what it is you're foraging for. You should know the lookalikes. You should be certain of it, but don't lie awake at night fearing mushrooms. Okay? If anything, if you want to go on a risk-based thing, lie awake at night fearing plants. You have a much greater chance of poisoning yourself or killing yourself from eating wild plants than you ever would from eating wild mushrooms. And I would suggest wherever you are right now, you could probably go out and eat every single mushroom and plant you come across. And at the worst, get an upset stomach. If you're intent on killing someone with a poisonous mushroom or a poisonous plant, you have to really go out and search for them. Not that I'm encouraging you to do that. Okay. So what are those fungi doing out there in the world? This is the second half of the talk. Be over in about 10 or 15 minutes maximum. I've classed what fungi are doing out there in 4 classes here: parasites, saprobes, predators, and symbionts. So parasites make their living from a live host. And here are a few we might see around. Sometimes the live host is us. So ringworm in the bottom right there is not a worm at all, but a fungal illness. Some of you may have fungi growing between your toes right now. No judgment. It simply implies that you're athletes, I suppose. The powdery mildews on the left are very, very common fungal parasites, especially on big leaf maple leaves. I'm sure a lot of you have seen that before and wondered what the heck. The fir blueberry rust is a parasitic fungus that alternates between any species of Vaccinium and any species of Abies. So on your island, most likely Abies grandis, the grand fir, and it produces 5 different spore stages. One of the spore stages on the firs can only infect Vaccinium species, and one of the spore stages on the Vaccinium species can only infect the true fir. It tends to make the Vaccinium species produce these sausage-shaped segments on the stem that have very few leaves and appear inflated. And we think it's just a way of making more surface area unobstructed by leaves to produce your spores on. And then a group of parasitic fungi that are called cordyceps. If you want to have nightmares tonight, I highly recommend you go to YouTube and look up zombie ants. And you can watch these ascomycete parasites infect ants and then control the movement of the ant. So the ant does what the fungus wants it to do: walk away from the colony, climb up to a high space, lock its mandibles onto a leaf, and only when all that's done will the fungus finish eating the ant and then produce its fruiting body through the ant's head, showering spores down on the other ants. Pretty cool strategy, but have a look at it tonight if you want to have nightmares. Generally in tropical forests, however, we have some of these zombie ant fungi. In BC, one of my favorite is Ophiocordyceps ravenelii. Here it is parasitizing a 10-line June beetle on the dunes near Ucluelet. The beetle is, well, you can see the beetle, and the two things that look like legs with knees on the left are the fruiting bodies of the Ophiocordyceps. It convinces, well, it doesn't convince, it takes over the function of the beetle, causes it to climb up on top of the dunes where no self-respecting beetle would ever be. That's where the predators are. And then when it's got it where it wants it, it finishes off killing the beetle, and it produces its pair of fruiting bodies. You can see them on the left here through the eyeball sockets of the beetle. Pretty cool, huh? Yeah. A little horrifying to some, maybe just fun for others. So here are the 10-line June beetles collected by a couple of friends on the dunes near Ucluelet. And you can see them kind of horsing around, but maybe considering eating them, because another cordyceps species is one of the world's most sought after aphrodisiacs. And so maybe that's what they're going for here with the 10-line June beetle one. Anyway, that's parasites. Saprobes simply live on dead hosts and decay them. A lot of our smaller fungi do that. So again, these are just the fruiting bodies. Remember, the fungus filaments are growing in the substrate and decaying the wood, decaying organic material in the soil, and so forth. One paper suggested that if we didn't have these and their bacterial and insect counterparts working on decomposing material, wood and material in the soil, it would probably only take a few decades before the organic material was piled up so deep that it would be as tall as the treetops and the entire system would grind to a halt. So these decomposers are important, and there's lots of them in our forested and grassland ecosystems. Here's the red belt conch. I'm trying to divide things into parasites and saprobes, but based on whether they have a live host or a dead host. But the red belt conch will start growing on a live tree, eating that live tree, and then when the tree eventually dies, it will continue to feed on the dead tree or the log. So it's a parasite, it's a saprobe. When it's a parasite, it's generally eating the heartwood, which is considered to be dead in a tree, in a conifer tree. So if you're feeding on the dead wood in the center of a live tree, are you a saprobe or a parasite? I don't know. Doesn't matter. A good example of how things don't always follow the categories we put for them. There are fungal predators. Here are a couple of well-known predators. Here are a couple of other well-known predators. In this case, it's not the fruiting bodies that are the predators, but the microscopic filaments growing in the wood that are producing the oyster mushrooms and the witch's butter. There's the oyster mushroom in the bottom right. There's a micrograph of its filaments with dots of a substance called ostriatin on them that paralyzes small insects so that the fungus can then slowly decompose them and slurp them up. Similar things are going on with the witch's butter. There are fungi that microscopically make these beautiful little nooses like arthrobotres that when the nematode gets its neck through one of them very quickly inflate to trap the nematode. In zoophagus, the fungi produce these little adhesive pegs, in the literature sometimes called lethal lollipops. So there are predators among the fungi, but there are way more symbionts. And just very quickly here for the symbionts, I'm going to talk about 3 groups of symbionts, but there are way more types of symbioses, generally between a plant and a fungus. The example I want to give you is Western yew. In the early 1990s, some of you may remember there was a bit of a gold rush for Western yew in British Columbia, when pharmaceutical firms in the United States determined they could isolate Taxol from the bark of Western yew. And the harvest in British Columbia wiped out Western yew in some areas of the West Kootenays and southern Vancouver Island. So I should mention that it turns out, when people look closely at it, that every single plant species on Earth, from tiny mosses to the tallest trees, has fungi. Lots of different species living asymptomatically inside their leaves, inside their twigs, presumably feeding on the products of photosynthesis and providing some kind of benefit to the plant. In a study of European yew, they isolated 242 species of endophytic fungi from surface sterilized yew tree twigs, 2 of which produced Taxol in a defined medium. Turns out it's not the yew tree that produces the Taxol, it's the endophytic fungi inside the yew tree. Makes me wonder how many of the compounds we assume plants produce are actually produced by fungi living inside them. By the way, sterilized twigs, 242 species of fungi living in European yew. Talk about biodiversity. Okay, but I am going to talk very quickly then about lichens, mycorrhizae, and mycoheterotrophs. Lichens are beautiful. They're about 95% fungi, generally a couple of species of fungi, and algae or cyanobacteria. So they are essentially fungi. Trevor Gower described them as fungi that have discovered agriculture. I really quite like that because they go to great lengths to cultivate the tiny plants that grow in them. Okay, mycorrhizae. So more than 90% of vascular plant species on Earth have fungi growing attached to their roots. That root fungus partnership is called a mycorrhiza, which means fungus root. So almost all vascular plants do this. The earliest fossils of land plants have mycorrhizae. So it's ancient, it's widespread. The fungi attached to the roots of these plants expand greatly the surface area through the soil that's available to the plant for absorbing water and minerals over what you'd get from much coarser root hairs. So the fungus provides these to the plant photosynthesizes, sends sugars to its roots. That's your very simple mechanism of it. If you want to know a bit more about it, read or listen to TED Talks from Suzanne Simard, great BC scientist and a good friend, and appreciate that when we did a study in the Victoria watershed, for example, we found that older Douglas fir trees generally had more than a dozen species of fungi growing attached to the roots of each mycorrhizal fungi, and that these were attached to the roots of multiple trees. So it's not a tree and its fungus, it's a tree attached to every other tree and a lot of other plants in the forest by these underground networks, swapping water, minerals, sugars, and signaling hormones. So in these forested ecosystems, there's a lot more collaboration and cooperation going on than the competition we were all taught about. The other thing I'll mention about mycorrhizae in British Columbia, because it'll be important to a lot of you, I think, is that almost all of the large mushrooms in British Columbia are the fruiting bodies of fungi growing attached to the roots of trees. That is, almost all the large mushrooms in British Columbia are the fruiting bodies of mycorrhizal fungi. That's why if you cut down the trees in an area, all of the large mushrooms disappear. What has not been demonstrated, because it's not something we can actually do in a natural environment, is if you removed all of those fungi from the ecosystem, all of the trees would disappear. So both partners need each other. And the reason we have forests in British Columbia is because the trees have fungi attached to their roots. Just to demonstrate that there are lots of different kinds of mycorrhizae. The ectomycorrhizae are the ones that are characteristic of plants in the pine family, poplar family, birch family, and so forth. Most other types of mycorrhizae produce microscopic fruiting bodies, but the ectomycorrhizae that we find on most trees in British Columbia produce mushrooms as fruiting bodies. Think of any mushroom you value, chanterelles, king boletes, hedgehogs, doesn't matter. They are all the fruiting bodies of fungi growing attached to the roots of trees. Every large mushroom in the autumn is. And then just final thought here, mycoheterotrophs are these distinctively weird vascular plants like the ghost pipe, what a lot of reference work still call Indian pipe. They are achlorophyllous vascular plants, or vascular plants without chlorophyll. How do they make their own food, or how do they obtain nutrition if they can't make their own food? They don't have chlorophyll, obviously. Well, they have fungi attached to their roots in mycorrhizae, as more than 90% of vascular plants do. And those same fungi are attached to the roots of nearby conifers, and the fungi transfer sugars from the conifers to the mycoheterotrophs. So there are a lot of reference works that will suggest these ghost pipes are saprophytes, and the coral roots and the gnome plants and similar things are saprophytes. Well, plants don't do that. It's much more interesting, much more fun, and it involves our favorite kingdom of organisms, kingdom fungi. Okay, I've got a bunch of other ways that we use mushrooms here, including magic mushrooms, but I think I'm going to skip over those to allow time for questions and discussions at the end.
Well, I have sort of a probably not very scientific question. First of all, I thought your talk was absolutely excellent. I have all your other books, and I'm going to go get the mycology one tomorrow or the next time I'm at the South End. I want to know, is it actually true that truffles need to be cultivated at the base of hazelnuts? And is it true that they need a particular dog species to hunt them out? And for those of us who love truffles, are there other ways to cultivate them in BC?
We estimate maybe 60 or more species of truffles in British Columbia. But you're talking about the European species now, I'm assuming, Suzanne. And there are truffle orchards on Vancouver Island, the Lower Fraser Valley, and as of last year, the Okanagan, that cultivate the European species. They generally grow them on either hazelnuts or European oaks. Probably more commonly on oaks than on the filberts, but that both are used in BC orchards. It's a tricky business. You start with tree seedlings that have been inoculated with these fungi, and put them in the soil and wait 7 to 10 years to see if it works. And with some of the early orchards, it didn't. So you wait 7 to 10 years and nothing happens. However, there are productive orchards now. The fungi fruit under the ground, so you do need some kind of an animal to find them. Traditionally, in Italy and France, people used pigs, but there aren't many people who hunt truffles with pigs anymore because the dogs have better noses and are much less likely to eat the truffles when they find them. So yes, people use truffle hounds. BC has a number of remarkable truffle hounds now that are used in these orchards to find the truffles. I've had the honor to meet some of these truffle hounds. There's at least one orchard, this one on Vancouver Island, that still uses the traditional services of Annabelle, a Vietnamese pot-bellied pig, to find truffles.
Wow.
You asked about the sort of dog. I've talked to the people who train truffle hounds, and the breed of dog is not very important. It's the training that's important. So there is a breed called the Italian truffle dog. Lagotto Romano, I believe, is the proper name of it.
Yep.
I have a friend who's got one of those that's been trained as a truffle dog, but the best truffle dog I ever met was a pug who always sounded like he was about to expire from gasping for air, as pugs are wont to do, and yet was the most remarkable creature I've ever seen for finding fungi, cultivated and wild.
Good to know. Thank you so much.
You're welcome. Adam, you have your hand up.
I do. I have a sort of what I hope isn't too impudent of a question to ask that's been with me for a little while now. It's about convergent evolution between fungi and plants. So just an observation, a number of plants that tend to smell horrible, like death or carrion, in order to attract insects, especially in the arum family, or like the quite famous Amorphophallus titanum.
Yeah.
Famously stinky, famously somewhat tower or phallic shaped, as is captured in their name. Similarly, in the world of fungi, if you think of Phallus imputicus or the stinkhorns, there is a similarity in that you have a similar morphology and a similar life strategy in terms of attracting flies and other carrion-loving species. And I'm wondering about whether you see any convergent evolution there in the form and function, and if there's any explanation for that.
I think there's lots of examples among plants, animals, and fungi on groups that we know are not related at any point in their evolution, developing similar solutions to the same problem. And if you want to attract flies, you got to smell like shit. That's the way the word works. So in the case of the plants, they're trying to attract the flies for pollination. In the case of the fungus, they're trying to attract the flies and other beetles to disseminate the spores either on their feet or for the blowflies in their digestive tract to actually eat that gross stuff. And so I think, yeah, that would fit the definition of convergent evolution, of unrelated groups developing similar strategies, that is, being stinky to try and attract flies. I figured that you were heading for Phallus imputicus when you said you had an impudent question.
Sure. I was curious about the morphology as well, right? If there's a relationship between that and the tower-like shape of these species, right?
I think it's more the way people see it, because I can think of a lot of non-phallic plants that adopt the strategy of smelling like rotten meat or feces in order to attract flies. It just happens that Amorphophallus and, well, I suppose you could also argue for the spade and spadix of the swamp lantern, and the absolutely remarkably phallic structure of the common stinkhorn, Phallus imputicus. So maybe there is something about the phallic shape combined with that I can't imagine what it is right now, but there's all sorts of things going on, especially with fungi, that I can't imagine, Adam. And so just because I can't imagine what it would be doesn't mean it ain't so. It's a nice observation that if you haven't seen a stinkhorn before, it resembles male human genitalia so remarkably sometimes that the genus is Phallus, and it's in the family and class, or the class and orders, Phallaceae and Phallales. So they just had to keep putting phallus in every level of the classification. It's a remarkable thing. Anyway, yeah, good observation. I'll think some more about that, the importance of the phallus shape combined with the stink. And if I ever publish anything on it, I'll call it Adam's stinky phallus syndrome. Here we go. Mary says, thanks for the great talk, although a little put off my dinner based on the ant story. And Mary, I highly encourage you to have a look at, there's some great YouTube videos, including featuring David Attenborough with fantastic filming on zombie ants. That is really an interesting look into insect fungal interactions. Daniel has a question.
Yes, thank you. What a delightful talk. Thank you. I've carried Pojar and MacKinnon around with me in the woods for years. And so to actually make your Zoom acquaintance is wonderful. My question is rather more boring and domestic than Adam's. I'm sorry to not keep the thread going. But I'm curious, as somebody who has a desire to cultivate native species of trees and shrubs, I'm wondering a couple questions. One is, I know I'm familiar with mycorrhizal relationships and the sort of obligatory nature of them, that trees need fungi, fungi need trees. And my first question would have to do with if that is also true of shrubs, woody shrubs, and other species. And my second question, I'm just going to dump them both on you at once and I'll shut up. The second question is, are there any lessons in the cultivation of native species in mycorrhizae? Are there ways to favorably influence the presence of mycorrhizal relationships, or are the mycorrhizal fungi so ubiquitous that there's no point? They're just going to find the matching species anyway, regardless of what we do. So there's two questions. One is having to do with non-tree woody species or shrubs that might also require mycorrhizal relationships. And the second, is there anything we can do to maximize the effectiveness of mycorrhizal relationships when cultivating or transplanting trees and shrubs?
Sure. So for the first question, yes, absolutely. There are very few plants, vascular plants, that don't have mycorrhizae. The mustard family plants don't, aquatic plants don't, and then it's just odd species after that. So anything you can think of growing almost certainly requires mycorrhizae. And there are different sorts of mycorrhizae. For example, there's a group of mycorrhizae called the ericoid mycorrhizae, which are the mycorrhizal associates of plants in the heath family. Not all of the heath family plants, but most of them. So if you're growing, say, salal or huckleberries or something like that. And incidentally, ericoid mycorrhizae are particularly good at accessing organic forms of nitrogen, like proteins and amino acids that we find in the abundance in our soils around here, and making them available to the plants much better than other mycorrhizae. And that probably explains why heath family shrubs are so common in our forests. You know, why when you think of forest understories, you think of huckleberries and salal and false azalea and things like that. So yes, just about anything else you're going to think about growing requires mycorrhizae. And for the second part, it's probably not something you need to worry an awful lot about. So these fungi appear to be ubiquitous and easily available. The only really good examples we have of plants lacking their mycorrhizal fungi and suffering for it are from forestry. For example, when people will take Douglas fir seedlings to plant in Chile. And so there have been some Douglas fir plantation failures where you've taken Douglas fir and, say, as an example, and moved it somewhere on the other side of the globe. But around here, it really shouldn't be a problem. The only thing I'd be concerned about is if you're planting seedlings into anything that's been sterilized, any soil that's been sterilized by any means, then I would be a little bit concerned, but that would be a rare situation, I would think. And if you're at all concerned, what I encourage people to do is to simply grab a handful of soil from wherever you've got that plant and put it in the soil where you're going to grow your new plants. And you'll bring lots of fungi with it. Or if you've got hundreds of seedlings, maybe a shovelful of soil. But that kind of approach to it, there are products you can buy in nurseries that promise to increase mycorrhizal fungi in the soils. And there have been a couple of good studies done that document that oftentimes, I mean, if you can imagine, what are the chances that that bag's going to contain mycorrhizal fungi that are appropriate for the plants on Galiano Island? Well, that's probably pretty small. And the other problem with those commercial projects is that one study at least found that a lot of the material in the bags was dead. So you're much better off taking a shovelful of soil from where you got it than using any of those commercial products. Okay.
Thank you very much.
You're welcome. Shauna.
Hi there. So I'm trying to start a project where I embed some logs, some red alder logs, into a garden structure at our local community school, because I'm in charge of the garden there. And of course, I really want to try to introduce Pleurotus species into the log. And I mean, my basic idea is just to take the fruiting body and try to rub it all over it like little holes in the log and to hope that the spores take. But I was just curious if you had any suggestions on that.
Sure. Inoculating alder logs with Pleurotus is one of the most popular mushroom cultivation things all up and down the West Coast. And so my first suggestion to you would be to just have a look at videos. So just hop up on YouTube and ask for videos. And there are videos up there that show proper cultivation of it. People generally do a little bit more than that, in that if you can get older plugs, take plugs out and put the material right into the log, you'll get a more likely and greater take on it. You can the mycelium of the oyster mushrooms is one of the most easily cultivated on straw or newspaper or things like that. So you can build up a large amount of inoculum pretty quickly, and that will grow much better for you than simply trying to shake off spores from the caps. So that's how it's generally done, is to, in very low-tech fashions. There's very high-tech ways of doing it, ignore them. But in very low-tech fashions, grow the mycelium on something like straw or newspaper or some organic material until you've grown up a large amount of spawn. And then people have taken plugs out and just to get it further into the log and sealed them up. So just a few steps like that, I think, will increase your success. But it's one of the easier ones to grow. And I think that oyster mushrooms are one of the most delicious of the mushrooms. And I also think it's very fun too, that as I mentioned in the talk, their mycelium produces this substance that paralyzes small creatures, which it subsequently eats, which means that the oyster mushroom, which is a staple of the vegetarian diet, is actually a carnivore itself. But anyway.
That's great. Thank you.
Okay, you're
The
welcome.
kids will enjoy that. I'm sure they'll enjoy that fact. I'm going to share that with them.
Okay, good. Thanks very much. Okay, Pearl has a question.
Oh, hi, Andy.
Hi, Pearl.
I'm really happy to meet you too. I have your book here. I wish you could autograph it. I've clutched it for, I don't know, over 20 years. It's a
You don't want me
1994 edition.
You don't want me to autograph it today, Pearl. I tested positive for COVID this morning.
I know, I heard that.
Let's hold off until my quarantine's over.
I'll wait till you're better.
Sure.
And I hope it's not too bad for you. I have a question about the extreme weather, like the heat dome and the long deep freeze we had last winter, and how that affects our local fungi. I know it's really done havoc with my garden. Some of my plants, some of my flowers wouldn't bloom, and some of my shrubs died, and other stuff is doing well, I think, from the rain. So what do you predict with climate change and weather changes? What will the impact be on these species?
Well, there's going to be a lot of changes, and we've seen some of them already. For example, we've seen increased tree death with Sitka spruce near the eastern end of its range, near Souk. There's been a lot of Sitka spruce death. Western red cedar and Douglas fir on the east side of the island and the southern Gulf Islands. I'm not sure exactly what you're seeing on Galiano, but I was involved with some people in Pender who were monitoring Douglas fir death, and there were a lot of trees dying about 2 years ago. And of course, when those trees die, the fungi associated with their roots die. Some of the fungi are associated with the roots of multiple tree species, and some of them with only one tree species. And so there's going to be a real shift in the mycorrhizal fungi associated with the roots of trees and shrubs also. We're seeing there's a distinctive group of fungi that we know from, of mushrooms that we know from alpine ecosystems. And we've been watching tree line increase in coastal British Columbia and southern interior British Columbia as well. That is the height, the elevation on mountains that trees grow up to. As the climate warms, tree line has been increasing, alpine has been shrinking, and so habitat for alpine fungi has been shrinking. So we're seeing reduction in habitat like that. Of course, it's compounded by a lot of human caused problems as well. So for example, the primary reason we have so much less old growth habitat than we used to have has nothing to do with climate change, or very little. That's more human activities. But I'm not a great prognosticator, but I think I'm safe in saying that the main thing we will see is stuff we don't expect. The main thing we see, especially in organisms like fungi that live in such intimate association with ecosystems that are so poorly understood on many levels, that we're going to be surprised by lots of stuff. Just as an example, and not a fungal example, I was working with forest researchers this was probably 20 years ago, trying to figure out why yellow cedar were dying at elevation along the north coast of British Columbia on Haida Gwaii in southeast Alaska. The reason they were dying is because their roots were freezing because of global warming, which doesn't seem to make an awful lot of sense. But yellow cedar has roots that are particularly close to the soil surface, and so are dependent upon a thick winter mantle, especially at higher elevations, a thick mantle of snow to protect their roots from unseasonable frosts. And so as the temperatures were increasing, the snow cover was decreasing, exposing the roots to these rare frosts that come along. And so global warming was causing them to freeze to death. Like, who could have predicted that? And I suspect most of what happens with fungi and climate change is going to be that kind of a surprise. The forest pathologists are really concerned that a number of the fungi which kill trees are beginning to move north out of Washington and Oregon, species that we haven't seen in British Columbia before, and species of insects as well. We'll probably see more introduced species of all sorts in the province, but who knows?
Thank you.
You're welcome.